Hello，若刚你在吗？
哎哎哎，
哎，Hello，
我在，
你的这个 slide 怎么样？run 的可以吗？可以 run，
可以，我
可以。
是你们能看到吗？
那你是我可以看的。
OK OK，那应
你有
该就
video 吗
video 我也跑了一下，应该能看到吗？
？OK OK，能看的，能看的，
OK，
没问题。
那应该
OK，
没 OK
好了，好好好好好好，
OK。
好嘞。
Hello, everyone.
Oh,
Is Hang here.
yeah.
Okay.
Do we start or should we wait a little bit?
I think wait a few minutes. Right now
Okay.
it's the social time. You guys can chat.
Yeah.
Okay.
Okay, the social time.
It's the social time. I'm living in my basement right now because I just moved over to Maryland, so I don't have a really like a house to stay in. So
Oh, that's good. Maryland is good.
So when did you start? It September or like August?
August 1st. I started August 1st. It's
Okay.
like yeah, but I still like travel back and forth between here and New Haven. Yeah. So and because I don't have a lot of stuff here and I have adjunct position at Yale, so I can do some experiment over there. So
Okay.
I just got like rented the temporary place over here.
I see.
And yeah, and tomorrow like the weekend we have a hurricane in Maryland, but I'm safe, I think.
Oh, but you're in the basement, so you're
Yes.
safe.
Yeah, I'm in the safest place.
Cool.
How was your trip, Zhi, to Ohio State?
It was good. It was good. Yeah, I think I don't know if Yutong is here. It's a good place. The big lab space. The quality are very nice.
It was a nice talk. Yeah, congratulations.
Yutong is here. Yeah, thank you, Yutong. So, Ivan, so the Maryland
Yes.
has a different campus, one campus.
Two campuses. They have three campuses, I think. But like two major campuses. One in College Park. It's focus on the like it's undergrad students, most of them, and in Baltimore, it's a School of Medicine. So yeah, it's two campuses. But
So
we're kind of established a new biomedical engineering satellite school in the School of Medicine. Mark Gladwin initiated that, so
Okay.
which is good. Yeah.
So you're located in Baltimore then?
I located in Baltimore. Yeah, Yawen said there are three campuses.
I
There's
see.
a UMBC. She knows more. She's been here like she was been here like for longer, like 5 years.
I see.
So we can wait like 1 or 2 minutes, then if you can start it, introduce.
Okay.
I think we have
Great.
good number. We have 40. Yeah, that's good number.
That's great. Okay, let's get started. So let me introduce Dr. Ruogang Zhao. I know Dr. Zhao quite well, not only because we are from the same hometown, but also like he's one of the few bioengineers that regularly funded by big funding agencies such as NIH, NSF, American Lung Association. And he is one of the pioneers who developed the lung model to study fibrosis, which he's gonna give us a talk today. So Dr. Zhao, please take away.
Okay, thank you, Yifan, for the introduction, and also I want to thank the CALA committee for the invitation to give this talk. I'm very happy to share our work in the recent years. So let me this in the presentation mode. Okay, so can you all see my slides?
Yes.
Yes. Okay. Okay. Good. Okay. So so today I want to just give a talk on the recent year that we developed these engineered lung tissue models that in my lab for modeling pulmonary fibrosis and anti-fibrosis treatment. So on this title page, I show some of these interesting pictures of this engineered model that we created, as well as decentralized lung, and then the individual alveolars under stretching. So I'll provide details of these projects in the later slides. So I want to start my talk by introducing the mechanobiology aspects of the pulmonary fibrosis. So I know in this group, many people are from the biology side, the lung biologist, but I'm from the engineering side. So I want to just give a little bit background on the mechanical aspects of the pulmonary fibrosis. So as we all know that in the healthy alveolar walls are thin and compliant, allow this gas exchange. But in this fibrotic lung, this alveolar wall, they become thickened and stiffened, which inhibit this gas exchange. So this disease progression is mediated by injured epithelial cells, differentiated myofibroblasts, and then also the immune cells in a very complex mechanically activated microenvironment. So for example, that we know in this fibrotic lung, there are highly remodeled extracellular matrix, and then there's some mechanical stretching, and then there was also a mechanical contractile force that was generated by this activated myofibroblast. So, so in terms of the preclinical fibrosis models, there's some limitations for this preclinical model, especially in terms of this modeling the mechanical aspects of the disease. So, for example, the animal model, the most commonly used bleomycin mice, they resolve spontaneously after a certain time. And then also, all the animal model, they have limitation to predicting human response under anti-fibrosis treatment. Commonly used cell culture models such as 2D culture model and 3D organoids, they're able to recapitulate some certain aspects of the cell-cell interaction, but they don't have the capacity to model and then providing readout of the mechanobiology that involved in this disease. And then more recently, there's advanced models such as the lung-on-the-chip models that was started. I remember a former CALA talk was on this lung-on-the-chip model. So it's building a membrane that's separating this epithelium and the endothelium layer, that mimicking this the interface between these two cell types. So this model has gained success in modeling certain lung-related diseases, but in terms of mechanical property, in terms of fibrosis, this model lacks substantial ECM component because the model is built on this polymer membrane that with the pores, and then also this polymer membrane is overly rigid for the cell component, right? So it's not a physiological stiffness. Right? So given all this limitation in the current model system, so my lab has a focus on technological development on this engineered lung tissue models that can be used for or more suitable for this fibrosis modeling. So for example, in the recent years, my lab has develop these fibroblast populated lung microtissues and macrophage and fibroblast co-culture microtissues that can be used to study the fibrosis mechanism, as well as for use in drug testing and then drug screening. And then we also have some work that I also want to show today on this decentralized lung ECM that also be used in these engineered lung tissues. And we have also incorporating this lung slice model, which is very commonly used by many researchers, into mechanical stretching system to mimicking the mechanical forces or mechanical stretching, the effect of that on this on the lung slice. But in terms of this fibrosis drug screening or treatment, I just want to briefly review the current state of this treatment on the pulmonary fibrosis. So our treatment for this disease is still very limited. So the figure to the left showing has been several a dozen of clinical trials that was conducted in the past 30 years to find a cure for this pulmonary fibrosis, but only 2 drugs received FDA approval, which is Pirfenidone and nintedanib. But these 2 drugs only can slow down the disease, and they're not able to stop or reverse the disease progression. And then more importantly, the mechanism of action of these 2 drugs are not fully understood. Right? So therefore, there's still need to find new drugs, and then also to improve the current drugs for anti-fibrosis treatment. So so our approach in terms of this modeling fibrosis and then anti-fibrosis drug treatment has been using these engineered lung tissues. So I want to start by introducing our engineered lung tissue by introducing this first model that was using these microfabricated micropillars to create these engineered lung microtissues. Right. So the core idea of this model is that we are observing this lung alveolar tissue, and then the lung alveolar tissue has a very large surface area and a very thin thickness, suggesting it has very high aspect ratios. So we wanted to model this very high aspect ratio tissue using this advanced biofabrication technology. So here we're using a technology called a microlithography, which is the same technology that people are using to create computer chips or cell phone chips. The only difference is that we are creating these chips in the clean room, but then later we're using biocompatible polymer called a PDMS. And so the PDMS is a material that also used in those organ chip and lab on chip devices. So we're using PDMS to replicate those patterns that are created in the clean room, and then to forming these arrays of micropillars that you see in this picture to the right. So, and then once we see the fibroblasts and extracellular matrix proteins such as collagen into a microwell containing these micropillars, these fibroblasts, they will self-assembly the ECM around them to forming thin microtissue that supported by the head of these micropillars. So in the next slides, I will show a video. So this video show here the microtissue formation process. So as you will see in this video, these fibroblasts, individual fibroblasts, they will contract, and then collectively they will compacting this collagen gel towards the center. And then because the center there are 8 micropillars, so they're providing mechanical constraints to prevent further compaction. So as a result, this tissue form as a square shape that hanging on top of these micropillar heads. So I'll play the video here. Okay. So as you can see, over the time course of 12 hours, these fibroblasts, they spread, and then they working on the ECM proteins around them, and then collectively they compact, and then forming a square tissue that hanging on these pillar heads. And then the picture to the right showing these are the SEM image of some of these microtissue shapes that we form by varying the micropillar design by putting them into different locations. So these design mainly just show the engineering effort or the engineering capacity of creating this tissue, but they probably don't have direct biological significance. So we also perform this fluorescent staining in this one of these design, which we call the 4 leaflets design. So we stain for the nucleus, showing these fibroblasts. They are quite densely packed or populated in this tissue, and then we stain for F-actin, and then we show that the mechanical boundary condition of these 4 leaflets actually causing the alignment of these fibroblasts in this tissue. You can see quite obvious diagonal alignment in this tissue. And then also this formation process, they highly compact the collagen, so allow this tissue to form very thin ECM layer, just as we expected, to mimicking this alveolar tissue. So the bottom picture showing the cross-sectional view of this microtissue. As you can see, the tissue has very large span but very thin thickness. So the aspect ratio is very high. So that's what we desired. That's what we want to model this the thin alveolar wall of the lung. And then we further integrate this microtissue array with a multi-well plate. So here, if you can see this, each of these bright spot in this 12-well plate represent one of these microtissues. So in each of these big well, in a 12-well plate, we have 8 by 8 microtissues. So together, 12 well, that we have 768 microtissues that is available for testing 12 different biochemical conditions or 12 different drugs. So, so we have used this microtissue model to model some fibrosis progression or fibrosis induction under TGF beta. So here we add TGF beta into this tissue model because this tissue contains lung fibroblasts. So we show that this under TGF beta, there's highly elevated expression of alpha SMA and pro-collagen. And then also because these micropillars are made of this flexible PDMS, so the elevated contractile force that generated by these myofibroblasts can be measured by the deflection of these micropillars. Right? So these micropillars serving as force sensors that can give readout of the contractile force of under this TGF beta treatment. Right? So we show that these over the time course of 6 days, there's a highly elevated contractile force in this TGF beta treated condition. So together, these immunofluorescence staining as well as the mechanical force measurements suggesting that we are inducing this tissue into a profibrotic state. And then we also measure the stiffness, right? Because remember at the beginning, the introduction that these fibrotic tissue, they become thickened and stiffened. So here we're using a mechanical stretching system to measure the stiffness of this TGF beta treated tissue. And then we show that indeed this tissue become stiffer. So it's 3 times stiffer compared to the non-treated control. And then also the compliance of this tissue become substantially reduced. So this is what the clinical observation that in the fibrotic tissue, this compliance reduced. So together, at this stage that we have a working model of a fibrosis model, and then we also are trying to model another sort of fibrosis symptom or phenomenon is called honeycombing. So honeycombing is a CT, is a phenomenon that can be seen as a CT image that's represented by this open air cyst in this CT image. So the cause of this honeycombing is by this the bronchus that was pulled apart by this attraction force generated by the surrounding fibrotic parenchyma. So we're using this microtissue model as you see here, that we see this microtissue model and then in this long format, and then we're using the micropillars to punch little holes on this tissue, and then we treat this tissue with a TGF beta And then what we observe is that these holes, they actually being enlarged under this TGF-beta treatment, and that was which is caused by this elevated contractile force generated by this differentiated myofibroblast. So if you look at here, this is the top figure shows this control untreated tissue where the holes are small, but then the bottom picture showing this TGF-beta treated condition, the holes are highly dilated or enlarged. And then if you look zoom in picture of this one of the hole, you see around the hole there's a circumferentially distributed alpha SMA positive myofibroblasts, suggesting these are the active myofibroblasts that generating traction force that causing the enlargement or the dilation of these holes. And so we believe this is a sort of mimicking the mechanical aspects of this honeycombing or the traction bronchiectasis effect. So having this modeling all this sort of the disease condition, the next step is we try to use this system for anti-fibrosis drug testing. So as a first step, we just want to use FDA approved drugs as a proof of principle. So here we use Pirfenidone and then nintedanib, and then at both high concentration and low concentration. And so the protocol is that we induce this fibrosis using TGF-beta, and then we adding these anti-fibrosis drugs at the same time of TGF-beta treatment. So mimicking a preventative treatment regimen. So what we show here is that this Pirfenidone at high concentration entirely block expression of alpha SMA and pro-collagen, so very strongly inhibiting these fibrosis markers. But this nintedanib at high concentration only partially inhibit the expression of these markers, and then the nintedanib and Pirfenidone at low concentration that have even milder inhibitory effect on these protein expressions. And then in terms of the mechanical property change, and then we show that in a, similar to the protein expression, the Pirfenidone at very high concentration is causing drastic decrease in the tissue contractile force, whereas the nintedanib at a high and medium concentration, they partially inhibit the contractile force in this tissue. And then similar trend was also seen in this stiffness. So the Pirfenidone and the nintedanib at highest concentration, they can cause reduction in the stiffness, but low concentration, they have limited effect in terms of lower the stiffness. And then we also try this anti-fibrosis treatment on this honeycombing effect. So here is a comparison. The top figure shows the TGF-beta treated without anti-fibrosis treatment, and then the bottom shows this is a TGF-beta and the Pirfenidone together treated. So it shows that this under this Pirfenidone treatment, indeed, this anti-fibrosis treatment is inhibiting the dilation of these holes, so suggesting it has the capacity to preventing this traction bronchiectasis formation. So that was our the first work out of this microtissue system, and later we had a bunch of follow-up work that we published. So for example, one of the work is that we combine this microtissue system into a mechanical stretching device, right? Because the previous work was all static, there's no mechanical motion, but because the lung, you always have mechanical motion. So we combine this with a stretching system. So here there's a video that I want to play that you will see this. We built in a vacuum chamber that was driven by the vacuum force, and then the vacuum force is driving this transparent silicone membrane to stretch the entire microtissue array.
I think, yeah, it's not open yet. Yeah, right.
Yeah, now it's working. So you can see the surrounding, there's a vacuum chamber that driving is a pull down the whole membrane, and then as a result, the tissue in the center is being stretched. So and then we look at this tissue before and after stretching, and then we found that this mechanical stretching is substantially enhancing for example, the cell alignment in this tissue as comparison. So here you can see this is a very highly diagonal alignment that form. There's some initial alignment in this tissue before stretching, but after stretching, they have become highly aligned. And then also we tested this anti-fibrosis drug in this stretch tissue. And then what we found is that this mechanical stretching, generally speaking, is elevating the baseline contraction force for all the tissues. So it's like they exercise the tissue into a mechanically activated state. But the anti-fibrosis drug is able to sort of lower this contractile force for these activated tissues. So in a similar manner compared to this static treatment, static culture. So far everything was using FDA approved drugs as a proof of principle. So what about the drugs that hasn't gained FDA approval, right? So we in one of the recent work that we tested is a drug that was currently still under clinical development. The drug is known as a KD205, which is a ROCK2 specific inhibitor. So we tested this KD205 in this fibroblast populated microtissue, and then we showed that this KD205 at high and medium concentration have this dosage dependent inhibitory effect on alpha SMA and pro-collagen expression. And then also in terms of the mechanical property, they also have a similar trend. Right? So the high and medium concentration of this KD205 has inhibitory effect on the contractile force as well as the stiffness, right? But the low concentration has almost negligible effect. And then we also are using this system to test some nanomedicine. So here, working with a colleague in my department, we developed this anti-miR-21 using lipid nanoparticle to encapsulating this anti-miR-21, and then using this as a drug delivery system to treat fibrosis. So we tested this LP anti-miR-21 in our microtissue model, and then we showed that this LP anti-miR-21 substantially inhibit this contractile force generation almost to the level of untreated sample. And so it's suggesting this formula, this drug delivery formula is quite potent in terms of inhibiting this the fibrosis effect. And also we show that this LP anti-miR-21 has a very strong inhibition on the microRNA expression of this miR. And then we also run some sort of animal model in comparison. So here using the bleomycin mouse model, and then we are using this drug delivery system to treat bleomycin treated mice, and then we show that this formula has strong inhibition on the fibronectin and collagen, as well as alpha SMA expression, and then the histological analysis that are validating our finding that this alveolar structure was largely protected under this drug treatment, suggesting this is a potential therapy for this treatment. So the animal, so the conclusion find in the animal study is consistent with the study, consistent with the result that was obtained in our microtissue models. So everything so far was a fibroblast tissue, right? So fibroblast only monoculture microtissue, and then because the fibroblast that's the main sort of contributor cells in fibrosis, so we started with that. But then recently we became interested in the involvement of the immune cells, such as macrophages, in the development of fibrosis. So in the past 2 years, we have been working on this co-culture model of human macrophages with human lung fibroblasts in this microtissue model. So the first observation that we started with, just by looking at this the histological section of fibrosis tissue, and so human fibrosis lung section. So we find that in this fibroblast focus section, that there's a highly aligned and then highly compacted collagen, and the fibroblasts. And then by performing this CD206 staining, which stands for profibrotic macrophages, we show that these CD206 positive macrophages, they're embedded in between these alpha SMA positive fibroblasts, and then also they forming this strong alignment between these fibroblasts. So these are indicating that these macrophages, they're probably under some sort of mechanical activation, and then they're sensing this remodeled tissue microenvironment, and then they're in the activated state that's causing their alignment with this with the fibroblasts and then collagens. And then we also perform this data analysis on the public available single cell sequencing data from the fibrosis patient, and we find there's a highly elevated expression of Rho-A and then alpha M beta 2 integrins in these macrophages from a pulmonary fibrosis patient. So suggesting together these are histological and single cell sequencing data suggesting that these are there's some certain level of mechanical activation in these macrophages in fibrosis. So so realizing that, so our first step is that we just want to co-culture. We co-culture these macrophages with this fibroblast tissue and see what happened. Right? So here I have a comparison of a monoculture fibroblast only tissue and a macrophage and fibroblast co-culture tissue. So the picture to the left showing these are the SEM image of fibroblast only tissue. So as you can see, as we previously see that these fibroblasts, they can forming this diagonal alignment just by themselves. This is because the fibroblasts, they generating contractile force under mechanical boundary condition, they self-align into this diagonal alignment. But then the picture to the right showing these are the co-culture. So you see that on this fibroblast tissue, there's a lot of these bumps. So these bumps are macrophages. And then what we can see from this picture is that these macrophages, they forming almost like well bonded to the fibroblasts, and then also they kind of spreading, and then they have some level of spreading, and then also their spreading direction align with the fibroblast direction. So we want to see if this alignment is real. So we perform immunofluorescence staining of our microtissue, and then we using confocal to measure the collagen fiber, and then also measure the alpha SMA expression, and then the CD206. And then so amazingly, we find that these three signals, they in a highly aligned manner. So you can see here, these collagen fiber, alpha SMA, and then CD206, they highly align in this microtissue. And then also the CD206, these macrophages, they become this sort of star shape. They become a spreaded shape. That was a very interesting. So we want to make sure, because this we haven't seen this in the literature, and this is the first time that we have seen it. So we want to make sure this is not an artifact that just due to our the square shape of the microtissue. So because we have the engineering capacity to vary this design of the microtissue, so we created a different design just to validating, see if the macrophage, they still can align with our the collagen and fibroblasts in different design. So here we created two different design. One is a spiral pattern, another is the diamond pattern. And so as you can see in the spiral pattern, that these collagen fibers, they align along this the spiral, the main axis of the spiral. And then in the diamond pattern, these collagen, they align along this the edges. Right? So in both cases, you can see the CD206 positive macrophages, they do align with aligned collagen fibers. Right? So these very interesting, but at the same time, it's validating our finding that suggesting the macrophages, they do forming very strong alignment with this microtissue, suggesting they are probably sensing the underlying topology of these cells and the ECM, and then they become mechanically activated and then align themselves to this underlying substrate, the mechanical cues. So So we see this strong alignment, and then we want to see if this mechanically activated macrophage has any effect on the induction of fibrosis, or simply here any on the alpha SMA expression. So here we did a comparison. We had this fibroblast only tissue that treated with the TGβ, and then compare with a co-cultured microphage and fibroblast co-cultured tissue. So here we show that in this microphage and fibroblast co-cultured tissue, there's a highly elevated alpha SMA expression that almost to the same level of the TGβ treated case. But in this microphage co-culture, there's a CD206, but in this TGβ, because that was a monoculture, so there's no macrophage in that case. Right? And then we also measure the active TGβ secretion in the supernatant, and then we find this in this macrophage co-culture, there's an elevated level of active TGβ, suggesting is probably the paracrine signaling that was secreted by this macrophage that causing the differentiation of this activated fibroblast. And then we also measure the mechanical property that we show that this co-culture of macrophage, they do causing the elevation in the contractile force in these microtissues. And then we sort of took some of this high magnification image to look at the spatial relationship between these alpha SMA positive fibroblasts and the CD206 positive macrophage, and we show that they are in a very close proximity. They're almost side by side in this tissue, and then so suggesting that this paracrine signaling or whatever fibrosis signaling between these cells are probably occur in this very close physical contact manner. So, so having this model ready, then the next step again that we want to test this the response of this anti-fibrosis drug in this system. So here we are using Pirfenidone again. So we using Pirfenidone in a preventative treatment manner, and then also a therapeutic treatment manner. So in this preventative treatment, we are adding this Pirfenidone at the same time of adding the macrophage. So from the very beginning, we want to see if this Pirfenidone is able to prevent fibrosis formation. So here, indeed, this Pirfenidone treatment is entirely blocking the alpha SMA expression. But also, if you look at the CD206 staining here, there's a substantially reduced number of CD206 adhere onto this microtissue under this Pirfenidone treatment. And then also there's a drastically reduced secretion of TGβ in this treatment. So we also perform this therapeutic treatment. So therapeutic treatment is we allow this fibrosis to progress for 3 days under the co-culture of macrophage, and then we treat with a Pirfenidone. So in this case, we see that there's a partial inhibition of the alpha SMA, but still, again, there's a very substantial number of reduction in this adhere CD206. So both of these preventative treatment and the therapeutic treatment suggesting that this Pirfenidone is strongly inhibiting the adhesion of a CD206 positive macrophage onto this microtissue. So these, we suspect these relate to the inhibition of the mechanobiological or mechanoactivation pathways. So the next step is we look into some of the potential pathways that was affected by this Pirfenidone, because our previous single cell sequencing data suggests that this is the ROCK and Rho and then integrin that was involved in the mechanoactivation. So here we look into these two pathways. So here we stain for ROCK2. We show that this Pirfenidone at high concentration, they indeed inhibit this ROCK2 expression in this microtissue. And then because we also have this KD025, which is a ROCK2 specific inhibitor, so we also try this KD025 on this co-culture, and then we show that this ROCK2 specific inhibitor, they have a very similar inhibition effect on this ROCK2. Right? So together, this is suggesting that this Pirfenidone is probably acting. So the comparison between this KD205 suggesting that profibrotic is probably acting through this rock and roll pathway, inhibiting the mechanical activation of this macrophage. And then we also tested this the alpha-M beta 2 because that was again previously suggested. And then indeed this alpha-M beta 2 was strongly inhibited in this profibrotic treatment. And so together, so we believe these are the potential pathways that was inhibited by the profibrotic, and this was not previously known mechanism of action of the profibrotic, right? Because the profibrotic, it seems the previous report is mainly on this TGF beta pathway acting on the macrophage on the fibroblasts. So the effect of profibrotic on this macrophage, I think these are the some of the finding, the unique findings that by our study. And then also, I also just want to have some additional slides, introduce some additional work, other work that we have performed in the lab. So one is that we also did some of this decentralized ECM work. So this picture show here that we have previously acquired through a tissue bank of this sort of human donor, fresh human donor lung. Now super fresh. This is after overnight transport, so it's after 18 hours. But for ECM extraction, that's fine. So we perform this decentralization process protocol for 2 days, and then we wash away all the cells, and then we end up with this very pale ECM only lung, and then we digest it using pepsin into these bell inks, and then we're able to using this bell ink to forming hydrogel, and then also using them to forming this engineered microtissue using these bell inks. So here is a comparison of alkaline blue staining between a pure collagen, which is corneal collagen, purified collagen, versus a decentralized ECM form tissue. So you can see this decentralized ECM, they have very strong alkaline blue staining, suggesting there's a substantial amount of preserved proteoglycan that in this DECM. And then we also using the DECM as a non-digested format. So we just using a thin tissue slice of the decentralized tissue slice, and we reseeded lung cells, and then using that as a model for certain studies. So here it shows that we also integrate these the lung slice model with mechanical stretching because apparently most of these lung slice model are under static culture, or some maybe doing a perfusion culture, but the lung has mechanical stretching. So here we integrate this with the mechanical stretching, and then we show that we're able to perform this very high magnification sort of monitoring of the individual alveolar change under this mechanical stretching, and then so the picture to the right showing is a mechanical simulation. This is a finite element simulation showing there's a very heterogeneous stress and strain field in this stretched tissue, suggesting that these alveolar walls are not subject to uniform mechanical stress. So maybe some of the areas are subject to very high, some areas are low, right? So maybe the high area, the cells that in this high area are subject to a more mechanical injury, and that's where the disease initiated. And so these are areas, is currently ongoing project in the lab, and then it's under heavy investigation at the moment. And so with that, I'd like to summarize my talk. So my lab has utilized advanced biofabrication technologies to create lung microtissue models that can mimic unique disease features in fibrosis, and then also allow integrated biosensing of the mechanical aspects of the fibrosis tissue. And then the changes in the tissue mechanical, structural, and compositional properties during fibrosis progression can be modeled using our lung tissue, engineered tissue. And this disease model have been used to test the efficacy of anti-fibrosis drugs of different formula and at different stage of clinical development. So with that, I'd like to acknowledge the students, the previous current students who performed the work, my collaborators, and then also the funding source. And then I will be happy to take any questions. Thank you.
Thank you so much for your wonderful talk. And now it's open to questions. So I want to ask the first question. So like for your stretching, like we know the biophysical cues are important to, especially for maintaining the lung function and homeostasis. What do you think about the effect of the stretching on, is it on cells more or is it on extracellular matrix more?
So I think, you know, there must be a sort of multi-scale transfer of the mechanical strain from the tissue level to the cellular level. So, you know, for example, that we show here, that we just did a simple analysis that's showing, you know, the strain transfer. You know, if you apply a 10% strain on this tissue piece, it's not 10% that was uniformly transferred to every alveolar wall. It's you know, some are like, you know, 20%, some are like 30%. And so it depends on the geometry of your alveolar and then also the individual alveolar wall thickness. So the strain transfer can be very heterogeneous. And then the cells that are directly attached to that portion of the alveolar wall is probably subjecting to very different mechanical stretching level, right? So I think, you know, here there's some very complex biomechanical sort of issue or biomechanical phenomenon involved. So that's, you know, some area that it's we're very interested. So we're working on this to try to get this multi-scale resolution of these cells that under this mechanical stretching.
Thank you so much. Let's go to the chat. Dr. Liu Gang mentioned that embrace your work. So basically, he mentioned like he's confirmed his study on MIR-21 in the profibrotic in fibroblast. And
thank you.
Yan, do you want to ask about yourself, or I can ask on your behalf?
Sorry, my background may be noisy. So I'm just really fascinated by the microtissue system, and as they self-form like a sheet of tissue and probably also ECM. I just want to clarify, like, do you see a thick layer of ECM and how does the fibroblast distribute, or simply they're actually just a sheet of cell with like minimal amount of ECM? And on top of that question, I was wondering, like, if you are interested or you have already tried to kind of play the epithelial cells on top of this sheet and see what happens and how they differentiate or how they are maintained.
Thank you. Thank you for the question. Yeah, so the tissue that we created, the current version is, you know, for example, if you look at the cross-sectional view in here, this is about 50 micron thickness. And then the staining here, the green shows the F-actin, and then the red shows the collagen, right? So you know, it shows that these cells, they kind of in a layer, because 50 micron, the entire thickness is not that thick, right? So the cells, they're definitely not in just a 2D planar manner. So they're probably penetrating into this ECM a little bit, but together they forming into this sort of not exactly a 2D layer, but they penetrating, mixing or embedding with the collagen. But there's a substantial amount of collagen in here. That's for sure, because that's as we started with a micro well. So the collagen that we added is contained in the micro well, and those collagen were all sort of uptaken or all grabbed by the fibroblasts during this tissue formation. So the amount of collagen that we added, they're all here. They're all in this micro tissue. So there's definitely a substantial ECM in here. In terms of your second question, we indeed we have tried, you know, because we once we get this fibroblast only tissue worked, it just straightforward. The next step is to think that maybe we can add, you know, epithelial to it, add macrophage to it, right? So our, you know, the work we just show is the adding macrophage, but we also tried adding epithelials. So we tried adding just simplest, A549, and then we do see the A549, and actually we also tried MDCK, which is not a lung cell, but it's a very typical epithelial cells. So both the A549 and MDCK, they forming sort of compact monolayer of epithelium on top of this fibroblast tissue. So it's very interesting. I have this confocal that I am not showing here, but you can see on the confocal they separating this into layers. So that that's something that potentially that we can use to study this epithelial stromal interaction.
Exactly. Excellent. Thank you.
Thank you.
Ke Yuan asked a question under do you want to ask it yourself, Ke, or I'll read it out. Under co-culture with the fibroblast, why macrophage morphology look like spindle shape, kind of like the fibroblast?
Yeah, that's we are amazed by this result too, right? And then we at the beginning my first response is I don't even believe this data. I thought these are the staining that was got wrong by the students. So I asked them to confirm multiple times, but these are CD206, and then in the fibroblast channel you don't see similar morphology, right? So and then we look into the literature. There are some literatures. So if you're familiar with there's one group in UC Urban, Wendy Liu's group. So they using much simpler sort of engineer platform. For example, just the 2D substrate with the nano grooves. Right? So they're making these micro grooves into 2D substrate, and then they see the macrophage onto it, and then they also see the macrophage can sensing those micro groove and then become elongated. Right? So this macrophage become spindle shape or become elongated is not a something that people haven't seen before, right? But I think this our work is probably the first one that demonstrating this can actually occur in a 3D tissue environment. Yeah.
So basically, you mentioned like the follow-up question. I have a follow-up question on that. So like basically, you mentioned the patterning of your fibroblasts or whatever, like underneath of the macrophage, determine the organization of the macrophage on top. So basically to come back to the question I asked, so whether or not the cell signal or ECM signals is important for regulating the subsequent cell interactions. Like basically fibroblasts under stretch, they can produce a lot of extracellular matrix and they can form different patterns. And this ECM will generate different integrin and ECM interactions with the macrophage or other cells. So whether or not it's ECM dependent or is it fibroblast directly interacting with the macrophage?
Yeah. So that's that we thought about all these questions. So the integrin that we stain for alpha M beta 2, I believe that was a ECM, that was a collagen specific integrin that used by the macrophages. So and then we show that there's a very strong inhibition if you're adding Pirfenidone. So that was suggesting this is the macrophages, they're making direct contact with the ECM. And then also we have some the staining that for example here that we show this the side-by-side image of this macrophage with the fibroblast. I mean they're very close, but I'm not sure. I mean we actually stain for because I'm collaborating with Boris Hinz, who is very well known in this fibrosis field. So he has a previous publication claiming that this macrophage and the fibroblast, they have a CDH11 direct contact through CDH11. Right? That's how they making this a physical contact. But we stain for CDH11, but we don't see the signal. And then research literature is suggesting that this CDH11 that was in the mouse cells and then not in the human cells, right? Because our entire system is all human based. So we stain, we didn't see it. So whether or not there's other cell-cell junctions that was involved in direct contact between macrophage and fibroblast that we didn't look further. So our current data show that they at least they direct the macrophage directly make contact with the collagen fibers.
Thank you. Dr. Wei Guo Chen has a question, and he raised hand. And do you want to ask a question yourself? And he disappeared. All right.
Question is interested in your disease is lung, right? It's total lung. And then you seed your fibroblast into the disease lung, and then form alveolar.
Yeah.
You think just the fibroblast in lung can form alveolar?
No.
This is the first question.
You know, sorry, that may be misleading. That they didn't form.
Yeah, this one. Okay.
Yeah, that was the original alveolar ECM. That was the structure was maintained after washing, after decellularization, and then we just see the cells on top of that. So the cell just grabbing on the preserved ECM structure. And so the ECM still have those alveolar holes in them. Because
I see. Yeah. Right. Okay.
There's no regeneration.
Okay.
It's not regeneration.
Okay.
Yeah.
Another question is just follow Ranco's question. Yeah, the macrophages follow the morphological characteristics. It's look like the picture you showed on there is the macrophage look like the fibroblasts. You have a similar phenomenon, morphological character. So the structure look like it's kind of I would like to say it's kind of abnormal. I would like to say. Okay. Usually the macrophage is kind of is different. I would like to say it's kind of how to say irregular form. Okay. And another question is what is the mechanism the macrophage to contribute to the fibroblastic progress?
So at the current our data suggesting that was we did a certain characterization. We measured the TGF beta right in the supernatant, and we show there's elevated TGF beta. So at least one theory behind is paracrine signaling that the macrophage they secreting TGF beta, and then the TGF beta affecting the fibroblasts. So that's a potential, but whether or not, like I mentioned, whether or not they have a direct interaction, that we actually don't know at this stage. So maybe further testing is good. Yeah, and then also the morphology. For the comment, because we have no too much experience with the macrophage before, but we search literature. But like I mentioned, these kind of elongated spindle shape, it was reported in other study, but that was all engineered substrates, right? Micro grooved engineered substrates. So this is we're quite surprising that in this sort of 3D sort of tissue environment can also have this effect. But if you the histological staining, actually these macrophages, these 206, they also kind of elongated spindle shape, right? They're not like because our original thought there maybe they should remain as a like a flat egg or like a circular shape, but these are elongated, right? So it's promoting our initial interest because we see this picture that we thought, okay, there's some interesting stuff going on in this mechanical activation going on maybe.
Yeah, could be. Yeah. So do you know what is? I mean, it's just a second question is for the macrophage. What is the contribution for this macrophage for this fibroblastic progress? I mean, the fibrosis formation. You say the Rho or some superoxide or something like that. Or you add the macrophage into the fibroblast, and then the forms is how to say it? They promote the fibroblast to forms how to say it? Yes, fibroblast progress. I mean, so what is the contribution? I'm sorry.
You mean the contribution from
macrophage
to fibroblasts?
To fibroblasts, yeah. I mean that based on our current study, the number of assays that we did is that we only can say that we tested TGF beta. We tested CDH11, which didn't work.
Okay.
TGF beta is working, so maybe TGF beta is a potential mechanism. But but I'm not ruling out there's other possible mechanism, right? These things are quite complex. So maybe there's other mechanism that behind that we didn't even test. So based on our current testing, TGF-beta. But overall, the fibrosis is quite complex. Macrophage is probably just one of the factors, right? Maybe the epithelium, the injured epithelium, that also contributing to the process. So that part, again, we didn't test.
All right, thank you.
Thank you.
G has a question about the alpha SMA express. Some of the alpha SMA express CD206 myofibroblasts to macrophage or macrophage to myosin. G, do you want to ask the question?
Yeah, I think
these
are, I would like to ask, whoever knows, it looks like, that's multiple cell that co-express both, right? It's possible. I don't know. It's in this system or in vivo that myofibroblasts can express the macrophage marker, or macrophage could potentially trans-differentiate to myofibroblasts.
Yeah. That we are, because we working with the collaborator at McMaster University. What's the name? So they actually have a theory that these microphage, they may be expressing both the fibroblast markers, right? So what they call is microphage to mesenchymal transition, right? So microphage to mesenchymal transition. So at the beginning we thought this maybe that's what's going on, right? Because these cells they align so much, but then we closely. So that's this staining that we did, right? We closely look like multiple cell pairs. We just want to see this at high magnification. Look at these individual cells. They our final conclusion is not right. These are the ones that with the CD206. They only have CD206. They don't have direct overlapping alpha SMA. And then the ones that have alpha SMA, they only have alpha SMA. They don't have CD206. So we believe these cell type, they're still separate cell type. They don't have trans-differentiation between them, at least based on the marker that we look at it. Maybe there are some other markers, they have some trans-differentiation, but again, just based on the marker that the limited set of marker that we look at it, they don't have trans-differentiation.
Hi, Ruogang. This is Kay. Very beautiful talk. I just want a quick follow-up per the discussion related to the macrophage morphology. Have you thought about, before you co-culture with the fibroblasts, you just have some kind of dye to stain macrophages, then you could trace their morphological changes after co-culture?
Yeah, we did all that, the in vitro study, the 2D. So these macrophages are human PBMC derived, M0 and then to M2, and then we stimulating them using L4, L13. So on each of these steps that we did, immunofluorescence staining, and then we even conform our study just in 2D culture, just without co-culture. We are actually using Pirfenidone to treat the macrophage only in 2D culture, and then in those cases they also are inhibiting some of these adhesion of these macrophages. So we confirm in the 2D case these macrophages, they have this tendency, some of these spindle shaped tendency, but they of course in 2D you don't see their alignment, right? But they do have some of these sort of the morphology tendency in the 2D. So we have the data I'm not showing here. I didn't show here, but we in 2D data we did those validation in 2D.
So can I ask a question? Yin. Just a quick question. So you use human cell to do this culture. Is that possible to use the mouse cells, because there are so many knockouts? So they will benefit from your model to kind of interrogate those gene functions in vitro.
That's absolutely, that's absolutely, that's a straightforward that we can try mouse cells. But but at the beginning of this project, when we proposed to the NIH, the argument is that we argue that the mouse system, the animal system, they have limitation predicting human results. Right? So that's why all the current project is, the reason that we do in vitro model that we don't do animal model is because the in vitro model that we use in human cells. But of course, that's mainly for the argument of doing drug screening. But if for a mechanistic study, for all this knockout, as you mentioned, yeah, of course, the knockout cells that will be, directly, beneficial. If we are doing this model, we can directly see the effect.
Yes, absolutely. Yeah. Well,
yeah, that would be very straightforward to do that kind of
yeah. Thank you.
And Dusha have a question. Like, is active TGF beta secreted by myofibroblasts in your model?
We yeah. So so that's actually we stop at this picture. Right? So here, I think we compared 4 cases. So this untreated is the fibroblast alone. Right? So this untreated the fibroblast alone, and then this M2A is macrophage alone without fibroblast. So compare with this 2. So these 2 are all monocultures. So one is a fibroblast monoculture, another is a macrophage monoculture. So we show that this coculture has higher TGF beta than 2 of these monocultures. So suggesting that this coculture, they do have some elevated TGF beta effect. But but compare with added TGF beta, of course, there's no way to compare with this this added TGF beta.
And he has a follow-up question. What about TGF beta secretion in the in vivo IPF patient?
In
the IPF conditions. Oh.
I mean,
I'm not sure like what a human.
Yeah, I mean, TGF beta, that was confirmed one of the major factor or one of the major sort of soluble factors in IPF tissue. I think that's broadly confirmed. Many studies, they have confirmed TGF beta level is very high in IPF patient.
Can I ask? Sorry.
Yeah, go ahead. Can
I ask 2 simple questions? So the first one I want to ask, did you consider co-culture the different population, the subsets of fibroblasts? We know that we have a lot of different fibroblasts in our in vivo, like the pathogenic, the senescence fibroblast, and also different subsets of macrophages. Did you consider co-culture those, then check how they look like?
Yeah, I think our I mean I'm in the engineering school. I think the limitation of my research is to have access to high quality biological samples, and so that's why I'm working with the Yavin to get some lung tissue, all that stuff, just to have high quality sorted cells. That will be very beneficial to our research. I'm here. I'm open to collaboration. If anyone has that kind of cells, we're very happy to try. But for ourselves, we're in the engineering school. We don't have access to those subsets of cells. Of course, we're very interested. Because these macrophages, they are PBMC derived, but then I have always wanted to get this the sort of alveolar macrophages, from this the lavage. But then you have to collaborating with the doctor, when they're doing this procedure. I mean, we haven't been able to get those live alveolar macrophage. So those are very easy to implement in our system, but we have to have access to those high quality sample through collaboration.
Okay, thank you. And also, did you consider about whether you can co-culture with a cancer cell?
We have this model, this micropillar thing that we actually have A549 culture with the fibroblast before. So A549, they just simply forming a sort of black ball on top of it. So there's no clear morphology, but we have other NIH-funded program project that we to study this, the sort of cancer interaction with the T cells, but that was not using this pillar system, but was also fibrosis sort of centered question, the sort of cancer, tumor stroma, fibrosis stroma.
Yeah. And actually, I because your tech is very cool, and I also want to ask maybe very stupid question. I don't know how you tech support this fibroblast in your model. So I don't know whether you can capture the AT1 or KRT5, KRT8. This flat cell, they're also flat, right? They're also elongated. I don't know whether in your model whether you can capture those cells in your plate.
You mean AT1 epithelial cells?
Whether they can support in your model, like this tool can.
We tried A549. We tried A549 before in this model. They do form a monolayer. I mean, if everyone interested, I mean, I can show that additional data. It's not in my slides, but like I can find out. Just give me 1 second. I can.
Yeah, I just curious whether you can mimic the alveolar, how the alveolar
work. Yeah. We have that data. We have the A549. Let me see. Here. So let me share these data here. So if you look at here, so oops.
A549, I think I saw. Yeah, I saw it.
So yeah, so this is the tissue that we formed, this fibroblast tissue first, and then we added A549 on top of it, and then here we stain for this SPC marker, the epithelial marker, and then here is the view, the top view of after the A549. So you can see the morphology. This is a very different morphology from the fibroblast only. And so this is almost like epithelial morphology. The cells are very cuboidal, very densely packed. And then if you look at the cross-sectional view of the confocal, you can see that these are red, which are A549, they're all on top, right? And then these are green, these are the fibroblasts, they're underneath. So I think, this is suggesting these cells, they have a sort of separate layers of epithelial and the fibroblast and the stromal layers. They're because of the way that we see it is a sort of sequential seeding. And so we see the A549 later. And then we actually also did a bleomycin treatment. We showed that after bleomycin treatment, these A549, they all become, sort of disintegrated, the monolayer of epithelium is become disintegrated. Right? These A549, they sort of just floating away.
Yeah, very cool, very interesting. Thank you.
Yeah, so this is the epithelial cell. I think it will definitely work, but again, A549 is not the best model. If we have access to, I'm working with the Daryl Cotton, trying to get their alveolar sphere to getting those iPSC derived AT2 cells. If we can do that protocol in my lab, then we can see those cells on top of this. So
then you can co-culture with the alveolar macrophages.
Yeah, then that will be a tri-culture. That's very much more complex.
Thank you, very cool.
Thank you.
Oh yeah, Dr. Liu, like the second time he mentioned he loved your model, and he asked a question: is there any drug that did not inhibit SMA and fibronectin in 2D culture or a collagen contraction assay, but show effect in your model? So basically, it's like the conventional model is 2D, and now like compared to the 2D model, and whether or not your model has any better effect in terms of the response to some kind of drug.
I think the real value. I think the real value of my system is the sort of biomechanical aspects. Right. Can you still see it?
Yeah.
Okay. Yeah, but but sorry, what was the first part of the question? What's the 2D thing?
Yeah, basically he wants to say like whether or not compare with the 2D culture, for example, our current platform, and whether or not your model has any better response to some kind of drugs in terms of inhibiting SMA and the fibronectin expression.
I mean, better inhibition of alpha SMA and fibronectin.
Yeah, or other effect like on fibrosis phenotype.
I think I think it probably won't have better inhibition effect, right? Because if you talk about the better treatment effect of the drug, then you're considering the sort of diffusion limits of your model system, right? Because the 2D system, like people argue, the 2D system, there's no diffusion limitation, right? Because it's a thin layer. But most of the other 3D systems, such as the 3D organoids, because they're bigger, they're thicker, they're often several hundred micron thick. So often people argue they will have this diffusion barrier, right? So in those cases, the drug that you're added from the top layer might not directly penetrate into the deep core of those organoids, right? So that there's a difference, right, in between 2D culture and those 3D culture. But in our model, our tissue is so thin, right? Our model is only 50, 60 micron thin. So we don't have diffusion limitation. So our model, I think they have similar, in terms of drug penetration, in terms of drug efficacy, they have a similar drug penetration as compared to 2D. But I think the real advantage of our model is providing an additional readout compared to 2D, right? Because the 2D, you don't have this micropillar, you don't have mechanical readout, but we have this in situ real-time readout of this contractile force, stiffness, all that, right? So that, I think that's the main advantage of our model. But I think that's the main advantage.
But have you basically like do a single cell RNA sequencing or like a bulk RNA sequencing comparing the fibroblasts, for example, in your model versus 2D model in response to several drugs and under stretch or without stretch, and that will give you some kind of a gene target over there, and maybe not specifically on this SMA or fibronectin, but maybe other targets. Yeah,
we thought about that, but the cell number in our model is actually quite low. So in each of these microtissue, there are about like 300 cells in there. So if we do the entire device, we have 8x8 tissue. So sometimes some tissue fail. So we maybe end up like a 30, 40 tissue that you still have just several thousand cells to work with. That's quite challenging to run any sort of analysis on it. Right. Western blot, definitely you don't have enough. Single cell PCR maybe, single cell sequencing, quite challenging. So it's the cell number. It's a very small amount.
Yaman mentioned she can send you more stuff like iPS derived 82 cells and also alveolar macrophage from patient BL. Okay. So yeah, she's opening a tissue bank or something. One question I have actually is like there's no question, but the one question I have is that like how do you see your model as compared to human or rat lung model in terms of the kind of because your stretch is 2-dimensional stretch rather than 3-dimensional stretch, and also like the stretch on the alveolus is different in direction. Because it's not like a homogeneous stretch. So can you control different kind of areas? And that's one thing. And second thing is like during IPF or COPD in different LDs, those are heterogeneous disease. So it's not like in the human patient, it's only like it's not like some in the human patient, the fibrosis is happening homogeneously throughout the lung tissue. So My question is that how do you control your model to kind of model the disease like heterogeneous areas of the fibrosis? The third question is that during fibrosis, the stretch is definitely different from the healthy patient. So because also during development of the fibrosis or after you got fibrosis, all this stretch are different. How do you change or modify the stretch dynamics during culture? Those are the questions.
So that's you're hitting on all the major points in my NIH proposal, right? My recently
I never read it.
My recently funded NIH project that is trying to answer all that questions. Exactly. So for example, your second question about this heterogeneity. So we know that in the lung, in the fibrosis lung, the fibrosis progression is actually has a progressing from the periphery to the center region, right? So and then also from the bottom to the top. So there's a spatial progression in the lung. But then and I think a couple years back there's a Cell paper that by this the what name Ming Tang right. So the Cell this study this sort of differential amount of the stretch on this alveolar epithelial injury, right? And then how is that contributing to fibrosis? So we actually sort of interested in a very similar question, right? So exactly like you said, this heterogeneity, these slides that I'm showing here doesn't reflecting any heterogeneity, but we have a plan in this project that we have creating sort of a different level of stretch in these tissues. So hopefully, in one tissue that we can creating different level of stretch, and then observing, sort of study how this different level of stretch that affecting this sort of the disease progression. And then also your first question, the stretching that we're doing here is a sort of planar stretching, but in the actual lung, it's more like a three-dimensional expansion. I think that's, I have to admit, that's some like a limitation that our current setup. We are trying looking into this possibility of doing some more sort of expansion based, but then expansion based, then you also run into imaging problem, right? Because right now, these 2D stretching that we can actually take in movies as we stretch, right? So this really give us advantage of observing real time how this sort of alveolar change. So 3D expansion base that'll be difficult, but I admit this is a sort of 2D system always have these kind of limitations. So that's something that probably we have to address or improve in the next stage. And then what's your third question again?
Third question is basically the fibrosis dynamics. So it's for example, if you want to culture fibroblasts on decellularized lung slices or something, that would be different from it's different from like a fibrosis in development.
Yeah,
right?
yeah.
So how do you solve that question and using
so that then we then these the sort of when you stretch this lung tissue, then that's the base tissue slice that you start with is becoming important, right? Because there's a previous study show that they have this lung tissue, decellularized lung ECM, without adding any biochemical, just seeding sort of quiescent fibroblasts on those fibrotic ECM, and those cells can become fibrotic, right? They become activated, right? So suggesting that this ECM laden or ECM sort of this biochemical signaling or molecules that was still preserved in those ECM, they can activating with the cells, right? So So one thing that we propose to do is getting this IPF patient tissue and make those slides, and then we reseed them with the cells versus the tissue that from healthy patient, or no, the healthy donor, which does not contain those profibrotic factors, and then see the cells, and then subject to stretching, and see if there's a difference. And so that's, I think that's something that we can study, sort of the pre-established disease condition and how is that contributing the further progression of the disease.
Xiaobo, sorry to keep you holding. So do you have questions?
Great talk. I almost forgot my question after your last. Sorry, it's been very late. No, I missed in the middle. Maybe you already explained that. So what's the motivation? You look at the macrophage, different kind of layout. You have the spiral and the different, like two different. What's the outcome, like consequence, if macrophage has different?
No, it just because we are engineers. So all if you have to say all these things that I present today, these are all artificial models, right? None of these is directly, except for the long slides. Everything that these micropillar based these are all artificial models. And then, so in some sense, many of these phenomena that we see here are on these artificial models, right? So because we see these, these slides, the square tissue that we see this alignment. So I personally at the beginning, I don't believe it, right? So I asked my students, can you, number one, repeat this, and number two, if you change the design, can you still see this alignment? Right? So that's the reason that we changed to this spiral pattern. I, just want to make sure these are not some, artifact that was introduced by this culture system. But then after we tried, we show that these macrophages, they do align with these fibers, the fibroblasts. So it's suggesting this is a real effect.
So your conclusion is that macrophages can align themselves based on the fibroblasts. Whatever fibroblasts tell them, they will just follow.
Yeah, I think that's what we show here.
Okay, thank you.
Thank you.
All right. Sorry to keep everybody too late, I think. All right, thank you so much, Dr. Zhao, for your presentation and answering all the questions. I think maybe you'll get a lot of contact to use your models.
Okay.
Yeah, and
Okay.
yeah, a lot of collaboration.
I'm open.
Yeah,
I'm very open.
Great talker. Great talker.
Yeah, great talk.
Okay,
everybody, good night.
Okay, thank you, everyone. Okay.
Great talk. Bye bye.
Okay, bye.
Bye.